I should not make any sound.
Hi, good evening, all. Hope you can clearly visible my screen and I'm audible to you. So, before starting today's class, I just want to ask you that do you need any additional classes? Do you need any additional classes to solve your doubts and all? Because today's class is 11, week 11, and we might not have another class, or we might have in the next week. But additionally, apart from that, if you want to have any discussion session from the very beginning to the very end, please let me know, then I will conduct a class. Okay, so today's class will be starting. If you have any specific doubt, please let me know, or else I will briefly start with question answer solve, and then we'll go over the summary class. Okay, so hello everyone, I'm Ananya Chatterjee. I'm a teaching assistant of the course Experimental Biotechnology. Course instructor is Professor Vishal Trivedi of IIT Guwahati. So we are at week 11 now. We so far has completed week 10 tutorial classes, and we have solved 9 assignments, and all the classes I have been uploading in YouTube links of this link, and the folders of all tutorial classes are getting uploaded there here. So today I will start with solving your assignment problem. So the first question is, what type of bond holds the complementary base pairs together in the DNA double helix? So we all know that hydrogen bonds between those base pairs are basically which is the main bond which holds the complementary base pairs together in DNA double helix. Okay, so the answer will be hydrogen bond. Which of the following is the correct order of stages in a cell cycle? So what we have been reading so far, at first G1, then S phase, then G2, then M phase. So S phase is a phase where DNA replication happens, okay, and G1 is a preparatory stage for S phase, and G2 is a preparatory stage for division, and M phase is where division occurs. So the answer will be G1S, G2 and M. The next question is, in which phase of I hope I'm clearly audible. I'm being a bit low, but yeah. So the next question is, in which phase of cell cycle DNA is duplicated? So as I was discussing, in S phase the DNA is getting duplicated, which prevents reannealing of single strands of DNA during replication. Okay, so during replication, reannealing of single strand, what do you know? Single stranded binding protein. So what this protein does, this protein bind to the DNA strand and will prevent it from coiling with another strand of DNA. Okay, so the answer will be SSB proteins. Next question, what is the purpose of the denaturing step in PCR cycle? What is the purpose? We have read about PCR. So what happens? We basically amplify certain size of DNA. Okay, we have double stranded DNA, and those DNAs are bonded, okay, hydrogen bonded together. So to replicate one part of the DNA and to replicate one strand and simultaneously another strand also, what you have to do, you have to separate those double stranded DNA into single strands. So the answer will be, the purpose of denaturing step in PCR cycle is to separate double stranded DNA into single strands. Next question, how much each denucleotides, like deoxynucleotides nucleotides you were adding to the PCR reaction mixture? So from the experiment protocol, you have to remember that the amount is around 200 micromolar. Okay, now we'll go forward to next question. What is the purpose of the final extension step in a PCR cycle? Options are to denature the DNA. No, this is not true. To amplify the DNA, to add extra cycles to the reaction, to ensure all single stranded DNA is fully extended. Okay, so the answer will be to ensure all single stranded DNA is fully extended. So we'll go forward to next question. Why is Taq polymer is particularly well suited for PCR? So you have been studied from your childhood that you are using Taq polymerase. So why is it so? Why we are using Taq polymerase, and what is the source of Taq polymerase? So we have read that from Thermus aquaticus we have got this enzyme Taq. So that Taq polymerase people have studied and then used for using it in the reaction of PCR. So what could be the answer? First option is it is highly temperature stable and remains active at high temperature. It is less expensive than other DNA polymerase. It produces longer DNA fragments than other polymerase. It is derived from eukaryotic cells. So the answer is, we know that it's very like Thermus aquaticus. This bacteria is very high temperature stable. Okay, so from there whatever enzyme we are getting, that is also very temperature sensitive because of that enzyme, right? So that remains active at high temperature. Now next question, what is the primary advantage of using hot stirred Taq polymerase in PCR? Options: it reduces the ending temperature, it enhances specificity of amplification, it eliminates the need of primers, it speeds up the extension step. So the primary advantage of using hot start Taq polymerase in PCR is it enhances the specificity of amplification. In PCR primer design, what is the significance of GC content? So when you will do these things practically, so whenever you will be designing primer, you will keep something in mind, that what is the percentage of GC in your primer length. So there is a significance of GC content, because we know as much as we are increasing GC, the melting temperature or the TM value or the annealing temperature is also getting increased. Okay, so as we know that high GC contents makes primer strongly bind, because the annealing temperature getting increased. So the answer will be this, strongly bind to temperature. So that's why this is significance. Calculate TM of a 18 base pair primer with sequence 5 prime this sequence. So there should be a formula by which you can calculate that how much GC percentage is there. So in the previous class we discussed that formula, that how much GC concentration is there, you have to calculate from there, and you can if you know the primer length, you will be able to calculate the TM. So after this calculation, the answer here they got is 50 degrees Celsius. So if you solve the problem, you will be able to find. So in last class we taught about that. If we go to we'll try with this. Yeah. Yes, so the formula is so you have to go through very carefully the slides. Okay, every questions and every concepts are being asked from the slides, you can find it. So So the formula is not written here. Okay, whatever. So there is a formula from GC percentage you will be able to calculate, and if you are using any software like Snapgene and all, you will be able to generate the melting temperature, like annealing temperature of your primer very easily. So yeah, now the number of DNA duplexes obtained from one DNA duplex after 6 cycles of PCR are, this will be, that formula is 2 to the power n. 2 to the power n means how much cycle? So 2 to the power 6 will be how much? 32. Okay, so the answer will be 32. When designing PCR primers, why is it so important to avoid regions of repetitive sequence in the genome? Because what will happen then, those repetitive sequence, if you have repetitive sequence in the genome, those repetitive, then primers will go and bind to those multiple genomic locations and leading to non-specific amplification. Okay, so that's why designing, whenever you are going to design PCR primers, you have to keep in mind that it's very important that you should avoid any regions which are very repetitive sequence in the genome, because primers may bind to multiple genomic locations leading to non-specific amplifications. So what is the main application of multiplex PCR in genetics and genomics research? Options are DNA sequencing, gene knockout experiments, simultaneous detection of multiple genetic markers, DNA purification. So what is the main application of multiplex PCR in genetics and genomics research? So because of the need of simultaneous detection of multiple genetic markers, we do multiplex PCR in genetics and genomics research. The length of the primers used for PCR should be typically between 8 to 24 base pairs. So we'll be so that's the last assignment you had from week 10. So today, next class we'll be discussing assignments from week 11, which is live now. The last date is tomorrow. Please go through all the slides very carefully to solve those answers, and if you have any specific doubt, you can ask me in today's class. Else that next classes assignments I will be solving in next week. Today I'll be just briefly discuss of Professor Vishal's slide in good lab practices. So an internationally recognized definition of good lab practices goes like this: good laboratory practice embodies a set of principles that provides a framework within which laboratory studies are planned, performed, monitored, recorded, reported, and archived. New Zealand and Denmark were the first to introduce GLP, or good lab practice, in 1974. United States was the next to introduce GLP in response to the poor scientific practices prevalent in US around that time. Why it's so important? An international economic organization, the Organization of Economic Cooperation and Development, published the principles of GLP in 1981 under the name OECD guidelines for the testing of chemicals that were internationally accepted. Good lab practices, precautions to do, not recommended action, data register. So we'll be briefly go through all those good lab practices, because this is the course where you have learned all the theories you will be going to practically import, and simultaneously you have to be prepared with your practical prerequisite also. So the good lab practice is one of those. So you have to be very precautious, you have to be very cautious that whatever reagent you are working with, whether it's acid, whether it's chemicals, how to behave with this, what you should wear gloves properly, you should wear your goggles, and what is the not recommended action you have to take care, and and you have to very much regularly register your data. So far we have discussed about different techniques which are very much necessary in your daily basis research work, which are electrophoresis, immunology aspects, ELISAs, and all, then molecular biology, cell biology, and chromatography. So now we'll be discussing a few research problem. Okay. So the first problem is scientists have isolated a old rock sample with DNA sample from dinosaurs fossil. They have isolated the DNA and did PCR amplification with random primers. Now they want to determine the size of amplified DNA. So this is a fossil, the rock, and then what they did, they amplified the DNA by PCR, and then they analyzed the size of DNA. Okay, so they have isolated the DNA and then PCR application with random primers. Now they want to determine what is the size of amplified DNA. So they can so what they do, they will isolate the DNA. So you have to understand how to isolate genomic DNA or plasmid DNA, and then you have to do PCR. For PCR you have to, because it's a very long DNA, so you have to do random primers so that the whole area gets covered. And then after amplification, what to do, you can do DNA sequencing. Okay, then we'll go forward to another problem. Scientists have discovered a transcription factor in skin cells binds to a set of gene X. Okay, so discovered a transcription factor in skin cells which can bind to a set of gene X, which is responsible for changing its color due to exposure to sunlight. Now they would like to identify the transcription factor from skin cells. Okay, read the problem very carefully. So what happened, scientists have discovered a transcription factor. So what is transcription factor? It's a protein, so it's present in the skin cell and that can bind to a set of gene X. So this there is a gene X where those protein will get bind, and because of this binding, the changing in color happens, and it also happens due to exposure to sunlight. Now they would like to identify the transcription factor from skin cells. Okay, so here is your gene. You have promoter, exon intron. This is the coding region. Promoter, where the transcription factor A is coming and binding. Transcription factor protein binds to DNA for modulating gene expression. DNA is negatively charged. So what they can do, so if they want to analyze that if the DNA is interacting with protein or not, they can do EMSA. Okay, they will be able to do DNA footprinting to know the exact sequence where the DNA is bound, where the protein is bound. Okay, so experimental design. Here you have DNA plus protein 1 plus 1 x plus y, and then DNA protein complex. So if your molecular weight is 1 plus 1, the total is 2 and xy, and so the name is Z. Now x greater than Z or x less than Z. So you can do gel mobility shift assay, as I was telling, EMSA. So this technique relies on the fact that a small DNA has a much higher mobility in gel electrophoresis than what is bound to protein. So here you have loaded DNA alone and here is the protein. So what happened, whenever you were adding protein with DNA, the shift you can see. Okay, again DNA alone and protein. So DNA is 100 kDa and protein is 25 kDa. So what will happening, that because of the binding of protein or binding of multiple protein, in the gel the DNA is getting upshifted. Now again, the results, experimental validation. So here positive autoradiograph show the positive binding experiments. So this is nuclear extract, this is the probe, and then positive autoradiograph shows that the positive binding of the experiment. So there are several experiments you can do. Again, you will add DNA, you will add protein and wash with elution salt. So whenever you are eluting your protein, the DNA will also come, and then you will run SDS page and agarose gel. So then only you run X and protein in flow through. Okay, so this is how you can do the experimental design. So if you are binding your DNA to a certain column, then the protein will also get eluted. Okay. Okay, so this is not where you are binding, you are letting the protein bound to the column, you are just letting the DNA bound to the column, and along with DNA the protein is also getting associated and coming out, and then you are running SDS page and agarose gel. Now this is the experiments you can do. You can do chromatography, agarose gel electrophoresis you can do, and then SDS page. Another research problem: Mycobacterium tuberculosis H37RV was treated with drug and it causes generation of oxidative stress inside the bacterial cells. The ligand responsible for this effect was isolated, and now PhD student wants to identify the adapted proteins from Mycobacterium tuberculosis H37RV to understand the signaling effects and associated molecular components. Okay, so you have a MTP cell and it was treated with a drug, and what it causes, it causes generation of oxidative stress inside the bacterial cell. The ligand responsible for this effect was isolated by a PhD student, and he or she wants to identify the adapter protein. What are adapter proteins? You have a receptor, right? And underneath that there are some certain proteins which are interacting with the receptor and helps in cell signaling. Okay, so those adapter proteins he or she wants to check from MTB bacteria to understand the signaling events and associated molecular components. So what they can do, there is ligand and then your MTB, so cell lysate, so here you will have ligand along with the adapter protein. So different experiments you can do. You have to check whether the like ligand receptor binding, along with whenever you will be doing pull down assay along with those receptor, another how like what are the proteins which will come out. You have to do some cloning, which recombinant DNA technology. You have to do ligand production, coupling to matrix, affinity chromatography, SDS page. So these are the approaches you can take forward. Then another problem: protease was cloned from Plasmodium falciparum, and PhD student wants to identify substrate peptide sequence to understand its role in parasite metabolism and to design potent inhibitors. This is peptide. So you can treat the peptide with a protease. Okay, and then the protease will cleave into different small fragments. So you have to do this protease production, then peptide synthesis, coupling to matrix, protease assay, affinity purification, and LCMS. So how you will proceed through electrophoresis approach? So so um, so after so it's cloned from Plasmodium falciparum and wants to identify substrate peptide sequence. So what will do, you will just you have to clone. If you don't have the clone of your protease, you have to do that, and then you will synthesize it, and simultaneously you have to synthesize a peptide also. Peptide, amino acid, from different amino acids, a peptide, a certain length of amino acids are called peptide, right? A polymer, like multiple amino acids are linked together by peptide bonds. So like this, and then you will act the protease with the peptide, and different fragments you will get, and that you can get like you can do sequencing, and then peptide sequence you can identify. So this lecture is just to make you realize that whatever techniques we have gone through, all will be very much important for your current research work and in future research work as well. So um, So this is so far. If I have any specific doubt, just let me know. Or if you want to have any another sessions, we can have additional sessions where if you have any specific doubt, we'll be discussing. Because so far all the lecture notes I have covered. But next class, please join, because next class I'll be discussing previous year assignments and this year's week 11 assignments, and also week 12 assignments both. So do join next class, and apart from that, if you need any additional class, let me know. I'll be happy to conduct. So next class will be next Tuesday, again 6 to 8. So do join at around 6. So our class goes on till from 6 to 8. So same for next class also, it will be same. But if you need any additional class, I will circulate the information. If you have very specific question from the day one, we can revise again, and we'll do that. All the best for your exams. Do well. Please go through all lecture notes by Professor Vishal, and also I have been uploading those also, and lecture tutorials. You can go through slides very carefully, because every questions and concept wise also you will be very much clear. So feel free to ask any doubt in discussion forum. Okay. Thank you so much. Today's class was a bit short, because if you have any specific doubt, you can ask me. I can cover. Else, we will meet next class with some assignments, and then your doubts. How much time you have to
Stay. Stay.
Yeah, yeah.
And then
You're done.
then you are leaving.
So morning class is supposed to be 6 to 8.
Hmm. ஹலோ
சார்
அண்ணா சொல்லுங்கண்ணா.
இது இந்த காப்பர் இருக்குல்ல, குவான்டிஃபைட் காப்பர்,
ஆமாம்.
அது வந்து பேக் ஆஃப் 100 இருக்கு.
குவான்டிஃபைங் காப்பர். ஆ சரி.
ஸோ அது வந்து 150 200 சேர்ந்தா 350 ஆயிடும். 300 பண்ணணும் இல்லை 400 பண்ணணும்.
பேக் ஆஃப் 50 கிடைக்காதுண்ணா.
பேக் ஆஃப் 50 இல்லைப்பா, 100 தான் இருக்கு.
சரி அப்போ அதை இதாக போட்டுருங்க, 100 போட்டுருங்க.
150 போட்டோமா?
போட்டுருங்க
இல்ல
150.
நான் வந்து பிரிச்சு கொடுக்குறேன். 150
அப்போ நாங்கள் எடுத்துக்கிறோம் இங்கிட்ட. நாங்கள் இங்கிட்ட எடுத்துக்கிறோம். ஓகேண்ணா, அதுவும் ஓகே தான்.
சப்ளை பண்றது வந்து நாங்க வந்து 100 பேக் சப்ளை பண்றோம்.
சரி சரி சரி.
கொட்டேஷன் நான் பிரிச்சு கொடுக்குறேன். பட் 150 தான் கொடுக்கணும். இல்ல 150 200 தான் கொடுக்கணும்.
சரிண்ணா சரிண்ணா, பரவாயில்ல.
என்ன பண்ணுவேன்? 150 பண்ணலாமா?
150 கொடுத்துருங்க.
சரி, அப்போ ஐட்டம் நம்பர் 3 காப்பர் வந்து குவான்டிஃபைட் காப்பர் 150
ஆ
மட்டும்.
ஆ ஆ
வேற
சரி.
எல்லாம் ஓகே. அது இது கோல்டு வந்து பேக் ஆஃப் 100 வரும். நான் வந்து இப்போ டிவைட் பை 2 பண்ணி அதுக்கு 50 இதுக்கு 50 பிரைஸ் போடுறேன்.
ஓகேண்ணா, ஓகே ஓகே.
நான் சப்ளை பண்றது வந்து பேக் ஆஃப் 100 தான் பண்றேன்.
சரிண்ணா சரி.
சரிப்பா.